Good afternoon, children. Good afternoon to all. 12th Biozoology, chapter Application of Biotechnology. From application of biotechnology,
No, that's a let's go.
Class. Hello. 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 Good afternoon, children. Good afternoon to all. 12th Biozoology, chapter Application of Biotechnology. From application of biotechnology, the previous class you studied about gene therapy. What is mean gene therapy? Stem cell therapy, stem cell bank, PCR in clinical diagnosis, application of PCR test. So all that is topic you studied the previous classes. Now today going to study about the topic that is called transgenic animals. What is called transgenic animals? What is called transgenic animals? What are the uses of transgenic animals? What is called biological product? What are the uses of biological product? Animal cloning. How to produce animal cloning. So all this topic, now we see the full detail. First we go for transgenic animals. What animals? Transgenic animals. In early days, selective breeding method. What method? Selective breeding methods are used. By using of this selective breeding method to improve the genetic characteristic. By using of this selective breeding method to improve the genetic characteristic of organism, either livestock or other animals. By using of selective breeding method to improve the genetic characteristic of livestock and also other animals. At the same time, based on the advancement of biotechnology, also, is possible to manipulation of genetic desirable trait. Genetic desirable trait. Not only the selective breeding method, and also based on this advancement of what biotechnology, also possible for manipulation or production of genetic desirable trait of organism or animals. Not only the animals are produced, any organism. Plants also produce desirable characters of plants and animals or any other organisms are produced based on the biotechnology, based on the field of or advancement of biotechnology. What is called transgenesis? What is called transgenesis? Transgenesis is a process. What type of process? Introduction of DNA. Introduction of DNA. Either the DNA may be the foreign DNA or exogenous. The introduction of DNA into the genome of animals, any animals. Introduction of the DNA genome of an animals. When the DNA is introduced into the genome of an animal, create and also the maintain stable heritable character. That process called transgenesis. Transgenesis is a process. With help of the process, introduction of DNA. The DNA may be a foreign DNA or exogenous DNA. The DNA which is extracted from what? Healthier organisms, right? Introduction of DNA, genome of any animals. After introduction of DNA into the genome of any animal, and also to create and the maintenance of stable heritable character. What type of character we like? The type of characters are produced with help of this technology. That is called what? Transgenesis. It's called what? Transgenesis. Foreign DNA. What type of DNA? Foreign DNA. Foreign DNA is introduced. The introduction of foreign DNA, that is called transgene. Introduction of foreign DNA, that is called transgene. What is called transgene? Introduction of foreign DNA. Foreign DNA is introduced. That is called transgene. The foreign DNA is introduced and also production of animals. By introduction of foreign DNA, what is produced? Animals are produced. Production of animals by DNA manipulation. By DNA manipulation, that is called transgenesis. That is called what? Transgenesis. What is called transgenesis? Foreign DNA is introduced. Foreign DNA is introduced is called transgene. Foreign DNA is introduced, that is called what? Transgene. When the foreign DNA is introduced and also production of animals by DNA manipulation, that is called transgenesis. That is called what? Transgenesis. Introduction of foreign DNA and the production of animal. Introduction of foreign DNA and also production of animals, that is called this, that process that is called this transgenesis. The transgenesis also is called genetically modified organisms. genetically modified organism. The other name of transgenic is diagram space. Transgenesis. Foreign DNA is introduced called transgene. The introduction of foreign DNA, that is called transgene. And production of animals by DNA manipulation. Production of animals by DNA manipulation, that is called transgenesis. That is called what? Transgenesis. And also is called transgenic animals. Transgenic animals. Transgenic animals also are called genetically engineered organisms. Genetically engineered organisms. What type of organism? Genetically engineered organism. Also is called genetically modified organisms. Genetically modified organisms. Modified organisms. All are same. Transgenic animals. Also is called genetically engineered organisms. And also is called genetically modified organisms. Genetically modified organism. Foreign DNA is introduced. That is called transgene. What is called transgene? Introduction of foreign DNA. Introduction of foreign DNA. Right? And the production of animals by DNA manipulation. DNA manipulation. That is called what? Transgenic animals. What type of animal? Transgenic animals. And also it is always called genetically engineered organisms. And also it is called genetically modified organisms. Genetically modified organism. Next we go for steps involved in production of transgenic animal. What are the steps involved for production of transgenic animals? Steps. What are the steps involved? Production of transgenic animals. Production of transgenic animals. Next we go for steps involved for production of transgenic animals. Transgenic animal. The first steps for production of transgenic animals: identification and the separation of desired gene. Identification. What is called? Identification and also separation. What is called? Identify the gene and the separation of desired gene. Healthy gene. Healthy gene. The first steps involved for production of transgenic animal: identification of gene and also the separation of gene. What type of gene? Healthier gene. That is called desired gene. Desired gene. Next, after the identification is gene identification, separation is over. The next that is called selection of vector. Selection of vector. What is called vector? The organism which is carrying the gene. That is called what? Vector. Selection of vector. The vector may be what? Vector may be virus or bacteria. Virus or bacteria. Here the virus are used. Virus are used. Either selection of vector or direct transmission. What type of transmission? Direct transmission of genes. Direct transmission of gene. Direct transmission. The first steps involved for production of transgenic animal: identification and separation of desired gene. Right, okay. The second steps, that is called what? Selection of vector. The vector may be virus or bacteria. Here, vector used, that is called this virus vector. It's called what? Virus vector. The next, that is called third steps, that is called combine the desired gene with vector. Combine desired gene. Combine the desired gene or healthier gene with vector. With vector. So desired gene. Combine the desired gene with the vector. The vector. After the combine the desired gene with the vector, introduction of transport vector into the cells. Introduction of transport vector. Introduction of transport vector. Transport vector into the cells. Either cells or tissues or embryo. Transport the introduction of transport vector into the cell or tissue or embryo or any other mature individuals. Mature individuals. Right? Okay. After when it is introduce the transport vector into the cells or tissue or embryo or mature individual, demonstration of integration and also the expression of foreign gene in transgenic tissue. In this way, many organisms are produced. What are the organisms are produced? That is called this either mice or rat, pig, cow. These all are transgenic animals. Goat, sheep, fish. These animals are produced. Cow. These all the animals are produced. The first steps which is involved for transgenic, that is called identification. Identification of gene. Separation of desired gene. After identify, separate the gene, healthier gene, then the vector is selected. The vector is what? Selected. The vector may be what? Virus or bacteria. For in this technique, vector is used. And also direct transmission, transfer the gene directly or through the vector. The vector, what's called vector? The vector, the organism which is carrying the gene, carrier, that's called what? Vector. Your virus is act as a vector. Through the virus only what? The genes are transferred. Then the combined desired gene with vector introduce and also transfer the vector into either cells or tissue or embryo or mature individual. When it is a transfer into the cells, tissue or embryo in mature individual, next that is called demonstration of integration and also expression of foreign gene in transgenic tissue. In this way, finally the organisms are produced. Some of the transgenic organisms are produced with the help of this technology: mice, rat, pig, cow, goat, sheep, and fish. These all the transgenic animals are produced. Here, the diagram is a desired gene. The human gene introduced into this what? Egg of what? Animals. Sheep, egg donor. Who's donate the egg? That's called egg donor. With help of this micro injection, the gene is injected into the egg of the sheep. Then the development, then microinjection of the human gene into the host organism. That's goat. Development takes place in the body of the goat. But next there is they produce a desirable organism that is called transgenic goats are produced. From this transgenic goat, they produce a what? Milk. The milk contains of what? Rich sources of highly rich sources of protein. In this way, the many organisms are produced. Transgenic organisms are produced with help of the technology. Mice, rat, pig, cow, goat, sheep, fish. These all the organisms are produced with help of this technology. Next, we go for transgenic uses of transgenic animals. Uses of what? Transgenic animals. What are the uses of transgenic animals? Uses of transgenic animals. Uses of transgenic animals. Many animals are produced. Transgenic animals are produced. One of the very important uses of a transgenic animal. Most of the transgenic animals are more efficient than this normal counterpart in feed assimilation. More effective. Most of the transgenic animals more efficient, not effective. More efficient than the normal counterpart when compared than other animals. Normal animals when compare than normal counterpart in feed assimilation. This point not given over above. You note it. And compare them these normal animals, the transgenic animals are consumption of more food. Consumption of what? Intake of more food. That is called feed assimilation. Most of the transgenic animals are more efficient than these normal counterpart in feed assimilation. Feed assimilation means consumption of more food, eating of more food when compared than other animals. Next is called faster growth. Exhibit what? Faster growth. Most of the transgenic animals are the growth is faster when compared than normal animal. Faster growth. Due to this faster growth and achieve marketable size sooner. Maximum size reach within short time. Maximum size reach within short time. Marketable size. Marketable size sooner within short time. The growth is increased and also the weight also increase. They exhibit faster growth. These points also not given on both. Most of the transgenic animal faster growth and achieve the marketable size within short time sooner. Meat quality is good. Mostly all the transgenic animal, the meat quality, the quality of the meat is good. Meat quality is good. Meat quality is good. They are resistant to certain disease. Power of the disease resistance. High disease resistant power. Most of the transgenic animals are resistant. What type of resistant to certain disease? Resistant to certain diseases. For example, for a arthritis like that. Certain disease. Certain disease. Most of the transgenic animals are helps for transgenic animals are improvement of some genetic characters in animals. Improve most of the transgenic animals helps in improvement of genetic character. Improvement of genetic characters. Improvement of what? Genetic. With help of these transgenic animals only, we improve the genetic characters of what? Characters in what? Animals. Characters in animals. And also the transgenic animals exhibit many transgenic models or exist for many diseases. Many of the transgenic animals are affected by the diseases. Many of the transgenic animals are what? Affected by diseases. What type of diseases? Affected by many diseases. That is called this what? Cancer. Cancer. When compare the normal animal, the transgenic animals are affected by the diseases like cancer, cystic fibrosis. What is called? Cystic fibrosis. Cystic fibrosis. Cancer, cystic fibrosis, sickle cell anemia. What is called? Sickle cell anemia. Many of the transgenic animals are affected by certain diseases like cancer, sickle cell anemia, cystic fibrosis, and also rheumatic arthritis. Rheumatic arthritis. What type of arthritis? Rheumatic arthritis. And compare the normal animals, the transgenic animals are affected by the diseases like cancer, sickle cell anemia, cystic fibrosis, and rheumatic arthritis. Transgenic mice. What type of organisms? Transgenic mice. The mice which is produced, the method of transgenic. The transgenic mice are used for testing of safety of vaccine. Mice used for testing of vaccine. Testing safety of vaccine. That's a very important uses. The transgenic animals is important for improving the quality. Most of the transgenic organism, improve the quality. Important for improving the quality. Transgenic animals. Important for improving quality. Improving quality of what are the product? Milk. Improve the quality of milk, meat, and also egg. And also eggs. Woolen product. What product? Woolen product. In addition to testing of drug resistance. In addition to the testing of drug resistance. These all the uses of transgenic animals. These all the uses of what? Transgenic animals. They exhibit faster growth. Most of the transgenic animals are growth is what? Faster. When the growth is faster, to achieve the marketable size sooner in short time, make market. Meat quality, most of the transgenic animal meat quality is good. Most of the transgenic animals are resistant to certain disease, not all the disease, certain disease. Transgenic animals are help for improvement of what? Genetic characters in animals. Next, many of the transgenic animals are affected by certain disease. Cancer, some of them are affected by the cancer, some of them are affected by sickle cell anemia and cystic fibrosis and also rheumatic arthritis. Transgenic animals like mice used for testing safety vaccine. Transgenic animals are important for improving the quality of milk, milk, eggs, and also woolen products. In addition to testing of drug resistance. Testing of what? Drug resistance. Next we go for biological product and their uses. What is mean biological product? What are the uses of biological product? First, we study about what is called biological product. What is called what? Biological product. Biological product, a substance derived from living organisms. A substance, what is called? Biological product means substance derived from living organisms. Any living organisms. Any living organisms. A substance which is derived from a living organism and used for uses of substances, used for prevention of either prevention, what is called either prevention or treatment of disease, either for prevention or treatment of disease. That is called biological product. Biological product means a substance derived from a living organisms. What organisms? Living organisms. And the substance used for either the purpose of prevention of the disease and used for treatment of disease. That is called biological product. What is called biological product? A substance derived from living organisms. And it is used for either prevention of disease and treatment of disease. That is called biological product. That is called what? Biological product. What are the biological products? Some of these biological products like what? Antitoxin, bacterial or viral vaccine, either bacterial or viral vaccine, blood product, what is called blood product, and a hormone extract. Hormone is all a bioproduct. Is all about bioproduct. Hormone extract. These all are example for biological product. One example for biological product or bioproduct. One is called antitoxin, bacterial vaccine or viral vaccine, blood product or hormone extract. These biological product, where it is produced, these bioproduct are produced from what? Produced from living organism with help of the biotechnology. These bioproduct are produced through the field of what? Biotechnology. Biotechnology in living organism or in living systems. What systems? In living systems. Some of the biological product, one is called antitoxin. Second is called bacterial vaccine. Third, viral vaccine. Fourth one, blood product. Fifth one, hormone extract. These all are biological product. The biological product are produced through the field of biotechnology. In living system. In what called? Living system. In living system mean what is called living system? The living system may be microorganisms. Living system may be what is called? Microorganisms. Living system means microorganisms. Whether bacteria, virus, or fungi, or plant cell. What cell? Plant cell. Or what? Animal cell. What is called? Animal cell. The biological product are produced with help of the technology, the field of biotechnology in living system. The living system means maybe a microorganism or a plant cell or the animal cell, right? The same time, recombinant technology. What technology? The recombinant technology also possible to produce bioproduct when the demand of bioproduct occurs. When the demand of bioproduct with help of this recombinant technology also is helpful for production. What technology? Recombinant technology. Recombinant technology. When the demand occurs for this bioproduct or biological product, with help of this recombinant technology also possible for production of various bioproducts. Right? Okay. Many types of bioproduct is approved to use. What are the bioproduct approved for use? Many types of bioproduct. Many types of bioproduct approved to use. What are the bioproduct is approved for usage? Approved for usage. One is called therapeutic protein. One is called what? Therapeutic protein. The protein which is used for treatment. Used for what? Treatment. That's called what? Therapeutic protein is approved for usage. Monoclonal antibodies. Monoclonal antibodies. Monoclonal antibodies. Third, that is called vaccine. That is called what? Vaccines. Biotechnological protein. Biotechnological protein. Biotechnological protein also is approved for usage. Hormones. Hormones are used. Hormones are used. And also the antibodies. What is called? Antibodies. The antibodies is a substance. Next is called what? Antibodies. The antibodies are substance which is act against what? Antigen. Which is act against what? Antigen. Act against the antigen, destroy the antigen, protect the body from disease. Protect the body from disease. That is the role of what? Antibodies. That is the role of antibodies. Antibodies is specific substance which is act against the antigen, destroy the antigen. The antigen will be what? Pathogen. What is called pathogen? Disease causing germs. Disease causing germs. The antibodies act against the antigen, destroy the antigen, and protect the body from the disease. Protect the body from the disease. Recombinant hormones. Recombinant hormones also produce. Recombinant hormones like what? What are the hormones? Insulin. What is called? Insulin. HGH. That is called human growth hormone. What is called? Human growth hormone. Recombinant vaccine. Recombinant vaccine. These all are recombinant hormone. Names of recombinant hormone. What is called? Insulin. Human growth hormone. What is called? Human growth hormone. That is called HGH. That is called what? HGH. That is called human growth hormone. Recombinant vaccine. Recombinant vaccine. These bioproducts are approved for usage. And also monoclonal antibodies. What is called? Monoclonal antibodies. Monoclonal antibodies. The monoclonal antibodies used for cancer treatment. What treatment? Used for cancer treatment and also heart disease. What disease? Heart disease. Name the bioproduct which is used for treatment of cancer. That is called monoclonal antibodies. And also the heart disease. Also it is used. And also it is used for organ transplant. What is called? Organ transplant rejection. Organ transplant rejection. Name this bioproduct which is used for rejection of organ transplantation. That is called what? Monoclonal antibodies. What is called? Monoclonal antibodies. At the same time, transgenic animals also used as a bioreactor to produce desirable protein. To produce what? Desirable protein. And also another one is called recombinant protein. What is called? Recombinant protein. Monoclonal antibody, one of the bioproduct which is used for approval, that is called monoclonal antibodies, and it is used for treatment of cancer, heart disease, and also the organ transplant. Organ transplant rejection. What type of rejection? Organ transplant rejection. And a recombinant protein. The recombinant protein like human alpha lactalbumin. Very important. Maybe ask you need side. Recombinant protein. Name this recombinant protein. The recombinant protein that is called human alpha lactalbumin. This all this is available today. It is used in this for that is called human alpha lactalbumin. Also available today. These all are approved by bioproduct for today usage. Next we go for animal cloning. Cloning of animals. Right? Cloning is a process. Genetically identical individuals or organisms are produced. Genetically identical organisms are what? Produced. This animal cloning. The animal cloning is a process. With the help of the process, genetically identical organisms are what? Produced. And also the genetically identical organisms or the individuals, they are produced a artificial method. What is called? Artificially. And also naturally. Another one method is called what? Naturally. In naturally organism by method of naturally, they are produced by the method of asexual reproduction without involvement of what? Both the gamete. This is called what? Asexual reproduction. Artificial method in the laboratory and also natural method in what? In asexual reproduction. Cloning in biotechnology. The help of the cloning, the biotechnology to create exact copy like a parent's, identical like a parent's copy of the organism are produced. Not only the organism, like copies of cells or copies of DNA fragments are produced. In this way, the first cloning animal, that's called what? Mammals. First cloning mammals, the name is called sheep produce. Developed by who's developed? Dr. Ion Wilmot. Another one scientist is called Campbell. It is called what? Campbell. When was produced? When it is produced in 1997. The first cloned mammal, name of the mammal is called sheep. And it is produced by Dr. Ion Wilmot and Campbell in 1997. Dolly, the transgenic animals cloned was developed by nuclear transfer technique. The nuclear transfer technique also is called totipotence. What is called totipotence? Totipotency. Development of entire organism. Totipotence means development, entire organism development is takes place. In this, the donor sheep, the mammary glands is a donor. So mammary gland also is called the udder cell. Udder cell is a what type of cell? Somatic cell. Udder cell is a what type of cell? Somatic cell. Somatic cell means what? Body cell. Somatic cell means what? Body cell. Somatic cell, that is the udder cell. It is a body cell. It's isolated. And also it undergoes for subjected for starvation for 5 days. Then udder cell, with help of this udder cell, not undergo normal growth cycle is not takes place. Then enter into the normal stage and become a totipotent. With help of the udder cell only, they cannot produce a new cloned organism. The same time, an ovum or egg, or egg cell was taken from another sheep. It's a donor. Okay. Either egg or ova which is selected from another sheep. From this nucleus is removed. When the nucleus is removed, that is called enucleated egg. What type of egg? Enucleated egg or enucleated ova. After removal of nucleus, the udder cell is called what? Udder cell and also enucleated ova fused together in cultured vessel. In cultured vessel, the outer membrane of mammary cell was ruptured. Outer membrane, the udder cell, outer membrane of the mammary cell was ruptured, and the nucleus is covered around the membrane, around the envelope of the nucleus. The outer membrane of mammary cell was ruptured, allowing the wall to envelop the what? Mammary cells. Then after the cultured oviduct tissue implanted after the fusion of what? After the fusion of udder cell along with this egg, then it is fused cell was implanted into the another sheep. Implantation, attachment of this fused cell into the uterus of the what? Another sheep. That is called surrogate mother. Then later 5 months is over. How many months? 5 months later. It is called 5 months later. The dolly was born. Right? Dolly is the first animal cloned of transgenic animal which is used for using of what? Somatic cells. What cell? Somatic cells. And also it is produced without the process of what? Fertilization. The cloning of animals. The cloning is a process. What type of process? Producing of genetically identical individual of organism. The genetically identical organisms are produced one of the method, either artificial method or natural method. Naturally, it is produced by the method that is called asexual reproduction. In the field of biotechnology, the cloning organisms are creating a copies, which is a parent, it is something like a parent. They are copies of cells or the same copies of DNA fragment are produced with the help of this technology. In this way, the first mammal produced, that is name of the mammal, that is called sheep, and it was produced by the scientist Dr. Ion Wilmot and Campbell in 1997. The dolly is a transgenic cloned organism, and it was developed by the method of that is called nuclear transfer method. What type of method? Nuclear transfer method. method. It is called totipotency. Totipotency means the complete development. What development? Complete development or the entire development is takes place. The entire development of organisms. The entire development of organism. The type of development is called totipotency. It's called what? Totipotency. Right? With the help of this totipotent development, potential of cell, development of cell, cells development, tissue development, organ development, then entire organ system development is takes place. Right? From the donor sheet, what is removed? Udder cell. Udder cell is a what type of cell? Somatic cell. What is called? Somatic cell. What is mean somatic cell? Somatic cell means body cell, which is determine the body character. Which is determine the body character. The somatic cell is isolated. Then it is subjected for starvation of 5 days. 5 days. Then other cell not undergo for normal growth with the cycle. They enter in it because it is enter in what normal stage and become a totipotence. Later, the worm is removed from another sheet. The worm is consists of what? Consists of what? Nucleus is present. The nucleus is removed from this worm. Then, after removal of the nucleus from the worm, it's called enucleated egg. Enucleated egg. The enucleated ovum placed in a culture vessel. Then, udder cell also is added. The outer membrane of the udder cell, the mammary glands, cell was ruptured, hollowing the ovum around the nucleus. This central nucleus are present. The nucleus is surrounded by what? The outer membrane of what? Somatic cells are covered around the nucleus, envelope the nucleus. Then fused cells are what? Implanted into the surrogate mother. Another sheet. After implantation is over, 5 months later, dolly was born. Dolly was born. The dolly, which is produced with the cloned dolly, which is produced with the help of only the somatic cells. Somatic cells, and also without the process of what? Fertilization. Without the process of what? Fertilization. In this way, the cloned organism, first cloned mammals, the dolly was produced. Okay, children, remaining part of the lesson we continue later.
biology வந்து நீங்க ஆப்ரேஷன் பண்ணிக்கிறதுல புரியுதா அது ஓடி 1 மணி நேரம் தூங்கணும்